Sabrina and I are, but Sabrina can introduce herself.
I am the Research Development Director at Taylor.
And I'm Adriana. I'm in the middle of nowhere. That's not true. I defended recently, and so I am in the process of getting foot out in the real world after being so long in academia. I don't know what's going to happen with my life, but for now, I enjoy Phage Phridays because I get to talk about phage, which I'm a nerd about. And then for those who are down there, I don't know anybody that has not met Joe, but Joe's an amazing guy. I met him while he was finishing, right? That was I think he was already finishing or finished. Yeah, I don't know if it was already finishing or finishing his PhD in the Davidson lab in Canada, and then he can tell you from there his story, how did he end up in UCSD. Can you introduce yourself, Joe?
UCSF.
Sorry, UCSF. You're right, UCSF.
Not all great phage work happens at UCSD, although a lot of it does, or in San Diego generally. Hi. Thanks for inviting me. Joe Bondy-Denomy. I'm an associate professor in the Department of Microbiology and Immunology at UC San Francisco. I started here in 2015 as a fellow after finishing my PhD in 2014 with Alan Davidson. UCSF has these positions where you can sort of start a small lab and kind of be a postdoc with a tech and a student of your own, and but also sort of have the responsibilities of running a lab. And then got hired as a professor in 2017, and I love it here. And our lab is focused on phages, obviously, and CRISPR and anticrispers, and now we're working a lot on new immune systems in bacteria that block phage and trying to understand how they work and how phages fight back. So that's us in a nutshell.
That is super cool. That is absolutely super cool. So I thought about 3 topics that I could ask you about. The first one was the cool discovery of the anticrispers, because I know you've told the story, but people here, many don't know the full story. So it would be great if you could recap that out.
Like the very at the start, how it came to be.
Yes. Like you, Pseudomonas, and killing phage and killing... My project in grad school initially actually was to isolate Pseudomonas phages from CF lung sputum samples. The Hospital for Sick Children in Toronto is a really famous place and a big CF clinic, and I had samples and I was supposed to isolate phages, and I'd never done anything like this before, and I kept sort of messing it up and contaminating things, and we weren't sure which phages were from which samples, and so Alan decided to downgrade my assignment to working with bacterial strains of Pseudomonas that had been isolated from CF and other infections in the environment, and actually just inducing their prophages. And I started to get interested in trying to understand what integrated prophages do to their host. So my project ended up being that I isolated a ton of different phages that had been induced from lysogenic strains. I plaqued them on various indicators and isolated them, sequenced them, and did cesium gradients and proteomics and all this different stuff. But then I started integrating these phages into a single strain, so I could actually do something that was a little more controlled, and that was taking a wild-type aeruginosa isolate called PA14 or PA01 and giving it different prophages and doing that, let's say, 50 times. So then I would have essentially an isogenic strain collection, each with a different prophage, and I could ask what changed about those hosts. And I was working mostly on superinfection exclusion, which many people here are probably aware of, how an integrated prophage changes the host to make it resistant to other phage infection. I was pretty into that, and I was keen to have that be sort of my PhD. And then I made a serendipitous observation, which was that when I met this grad student named Kyle Cady, he sent me a strain that had the CRISPR system knocked out, and I realized that in fact some of the phages in my collection that never grew on PA14 grew on the Delta CRISPR strain. At which point it had an endogenous CRISPR system that until then hadn't been known to be functional. And then I realized, well, some of these phages in my collection are naturally targeted by the CRISPR system, and in fact that phages had protospacers matching sequences to the natural spacers that were in the strain, and sort of a light bulb went off and said, okay, CRISPR works. And then the real shock came when a couple of the lysogenic strains of PA14 that wild-type PA14 had resisted these CRISPR-targeted phages by using CRISPR, but once I gave some of these strains of prophage, they became sensitive to the CRISPR-targeted phages, if that makes sense. So it became clear that perhaps these prophages were inactivating CRISPR, and a few months later, I found the specific genes that were encoded in these different prophages that were sufficient to inactivate CRISPR on their own. And so it was very serendipitous, very unexpected, but the observation was that, prophages normally make their host stronger. In this case, these prophages are making their host weaker. And that didn't make sense, and I thought it was wrong for a while, and then eventually things kind of aligned. And later we realized in the latter half of my PhD that these were anticrispers were encoding small proteins that directly interact with CRISPR machinery and turn it off.
Told you that story was super cool. I got to hear it here, but I think that the story is cool. So after that discovery, you started your own lab, and you continued working on that. So there's a lot more to tell about that story afterwards, which you have been working on.
Yeah. Go ahead. Sorry.
No. So we could start with the work that what's her name? Now I forget names. Adair did, which sounds super cool.
Yeah. So Adair is an incredible scientist. She took a big risk to join working with me right after I started. She had done all her rotations and was set to join a lab, and decided to do a fourth rotation with me, despite having offers from 3 great labs to join. And I didn't have pipettes or tips or anything ready, but she joined and had a great PhD. She's now a Miller Fellow at Berkeley in Jill Banfield's lab, and is going off to great things, I'm sure. But she was really interested in why there were so many different anticrispers encoded by essentially the same phages and pseudomonas, and ended up trying to set up experiments to compare them and ask which ones are strong, which ones are weak, and is there a better strategy for inactivating CRISPR or a worse strategy? And she ended up coming across a realization that led to a paper later for her, which was that phages seem to cooperate to deploy anticrispers, which may not make any sense, but we observed that phages that encode anticrispers, if there's not enough phage in the population, they might as well not have anticrispers. CRISPR will totally drive those phages to extinction or limit their replication. But once there's enough phage in the population, they can replicate well and inactivate CRISPR. And she designed some really beautiful genetic experiments to prove that they're essentially phages that were infecting and donating anticrispers almost as a communal good, even though the phages themselves would die, so they were unable to protect their own genome, but their clones or their kin can benefit from that work and later, achieve success. And that we sort of called altruism, although it's obviously not a neurological kind of altruism, but one that is probably just a necessity, because CRISPR can be pre-expressed. The complex is loaded with guides that will cut the phage genome. It's almost like a shark in the water with its mouth open. The phage is really doomed, essentially. The same goes for restriction enzymes. And, it's hard to produce these inhibitor proteins fast enough. And so even though the anticrispers are expressed very quickly and very robustly, it's still not enough to neutralize CRISPR right away. This is very analogous to how CRISPR works. CRISPR doesn't acquire a spacer from every infection. CRISPR probably just acquires a spacer when the phage messes up or stalls, and then it can acquire this DNA into the bacterial genome. So it's sort of the imperfections of biology. When you think about the details, you realize that nothing works perfectly every time. And so CRISPR really, the important thing for me was CRISPR is still a big benefit, even when there are phages with anticrispers around. It's not true that the phage will always win. CRISPR will win a lot of the time, and eventually, maybe phages will win. So it put a lot more ecological context around the project, and that sort of ecological thinking was brought to us by our collaborators back then, Edsa Westra and Seneca Van Hout here at Exeter, who really were the drivers of the ecological thinking, and they published a paper at the same time as us with similar observations. So it forced me to think of phages in a different light, besides just if it has gene X, it will win, and if it doesn't have gene X, it will lose. It's much more complicated than that.
That's still a fascinating story. So, but there are different types of CRISPRs, though.
Yeah.
The anticrispers that you have found you found them against a lot of the
everywhere. They're really encoded by lots of different phages, infecting a lot of different bacteria. And you're right, these different microbes have many different CRISPR systems. Pseudomonas is fine. It has type 1F, type 1C, type 1E, and type 4A, for anyone who loves the CRISPR nerd jargon. And then we started working on Listeria, which is a totally different gram-positive bacterium with different phages. And it has type 2A, type 1B, type 6A. And we started working on Moraxella, which has type 1C, type 1F, type 5A, etc. They all have these different CRISPR repertoires. And the long story short was that we developed new methods to find anticrispers intentionally, whereas before it was very accidental. And the combination of bioinformatics and phage genetics methods to find them, and we were successful at finding anticrispers in all these different bacteria, now extended into Enterococcus and Streptococcus and Staphylococcus, various mobile genetic elements, phages and plasmids and conjugative islands that encode proteins that inhibit very specific or very broadly against the CRISPR systems of their hosts. And these are all turned out to be different tiny proteins with different mechanisms of action. And in some ways, common regulatory strategies where they're made very robustly and quickly during phage infection. They're actually also then repressed by phage repressor proteins that we worked on, which is sort of an unexpected twist that phages, when they have such strong promoters to drive anticrispers, they need to turn them off pretty quickly. So you can picture a phage coming in, makes a fast-on burst of anticrispr protein, turns it off quickly, and if that phage doesn't die, it will then go on to, if it doesn't get cleaned by CRISPR, it will go on to replicate. If it does get cleaned by CRISPR, then the next phage might benefit from the work of its predecessor. So overall, we ended up with this model of commonalities, i.e. quick expression, quick repression, and synergy between phages. Those are the commonalities, but then the differences are that the anticrispr proteins are highly variable, the hosts are highly variable, and the CRISPR systems are totally different. But some common themes have emerged over the years. And that's sort of where we're at now with the Anticrispr story.
So, okay. That means that you have found a way to modulate CRISPR in one way or another.
That's right.
Which has a bunch of implications. But before we go through the implications, I have one question, and this is a question that I was asked so many times when I presented your work in Journal Club in my lab, which is, are these phages that have anticrispers, are they all lysogenic? And does that make sense? And like, how does, because the overall question was always, oh, but anticrispers, so the spacers for the CRISPRs have to be from phages that have had some contact with the bacteria for a while, because acquisition is not that easy and it's not super fast. And so the conundrum was always, oh, are these phages always lysogenic?
The answer is no, they're not always lysogenic. The best story for lytic phages that encode anticrispers came from Sylvain Moineau's lab up in Quebec. They found some Streptococcus phages that encode a couple of really interesting Cas9 inhibitors, one of which is super broad spectrum and inhibits like every Cas9 that's been tested, pretty much. And so they are encoded by lytic phages. We do see homologs of anticrispers we find occasionally on lytic phages. We have mostly found them on temperate phages and plasmids, more so because of how we look for them. The database of genomic space is far greater for prophages and plasmids than it is for lytic phages, just because of how, if you sequence a bacterial genome, you might get 3, 4, 5, 6 prophages along for the ride without even trying. So we have so much more prophage genomic space, and we actually use a couple of bioinformatics approaches that lean heavily on finding them on prophages. The reason I think they're common on prophages, and there's been a lot of nice work on this from Stan Bruns and Edzo Westra and other people in the CRISPR field that have shown that integrated phages can eventually incur autoimmunity, whether there's a perfect spacer match there at the start that could cut the prophage. That makes the anticrispr an essential gene that has to be there to keep the cell alive, or else CRISPR will cut its own genome. CRISPR is very stupid in that regard, and unlike restriction enzymes that don't cut themselves, CRISPR will gladly cut their own genome if the sequence matches. That sort of ignorance has made CRISPR a great tool and will cut anything it's programmed to cut. But for prophages, that's a problem. And even if there isn't a perfect match, because CRISPR is adaptive, it can acquire new spacers
this will probably eventually lead to autoimmunity. And so I think anticrispers are common, not just for phages to use to help them kill their host, but for prophages to help persist, because the symbiotic prophage looks just like an infecting phage. CRISPR can't tell the difference between a phage that's there to kill it and a phage that's there to hang out in the genome. So I think anticrispers are so common on these sort of elements that can be symbiotic or parasitic at different times, because there's mutual benefit. Anticrispers can be very helpful for the host in addition to the phage. So not always on temperate phages, but that's our bias more from a searching perspective. We've actually turned our attention to asking how do lytic phages deal with CRISPR-Cas immunity, and the answers have turned out to be quite different, actually. We're working on a handful of lytic phages that seem to resist CRISPR-Cas immunity, but we think do not use anti-CRISPR proteins. We think they replicate in ways that are unique to lytic phages, doing things that temperate phages just don't do, and that helps them get around CRISPR, and so they don't always need anti-CRISPR proteins.
Wow, again, this is cool. So it has a bunch of implications for everything, including phage therapy.
I had a quick question.
Even more excited. Go ahead, Sabrina. You had a question.
I was really interested when you were talking about the dumb bacteria.
Dumb CRISPR.
Dumb
Yes.
CRISPR. Yeah, I'm curious. When the bacteria it cuts itself, do you think it's another way to create mutations in the bacteria?
Yeah. That's a great question. There has been some discussion of this. Actually, the earliest evidence from this was from Peter Finneran in New Zealand. He had a really nice paper, I think in 2013, that showed that the Pectobacterium Cas3 system would self-target occasionally and make big deletions, or at least select for loss of large regions. That wasn't totally clear. We've worked a lot on Cas3 lately as an editing tool. But this is a very processive nucleus that makes huge deletions. And so there is some evidence from Peter and from others now that CRISPR enzymes will self-target, and that absolutely can lead to mutation in that region. Whether that's adaptive or beneficial or not, selection will have to decide. Probably losing huge chunks of the genome is not advantageous, usually, probably not. But it's certainly possible that this is sort of like a diversity generator, and sometimes this can be positive. There's also been a couple of papers that have suggested that CRISPR-inducing mutations in phages can drive evolution into new directions. So it is, you can think of these as sort of maybe as evolutionary drivers sometimes, but I would assume usually maladaptive to have huge deletions or chunks missing from bacteria or phage genomes.
This is so cool. So now my question two questions. The first one, which is modulating CRISPR systems for gene editing purposes. You have now a tool that pretty much allows you to modulate those CRISPR systems. Have you guys looked at applications?
Yes, absolutely. We have not in my lab, really. We don't work with human cells or mice because I really don't want to. I love working with microbes. My disclaimer here from a conflict perspective is, yes, I started a company called Acrigen Biosciences, like Anticrisper genetics, Acrigen, to explore some of the human gene editing applications for anticrispers in the human therapeutic space. Being in San Francisco, it's like a rite of passage that you have to start a company. And so they're exploring that. What I can say is like there have been a handful of papers, including one like yesterday in Nature Communications, showing how one of the listeria phage anticrispers we found can be used in gene drive applications in mosquitoes to erase the gene drive, which is absolutely amazing. Gene drives is a powerful technology that can be used to wipe out species in the wild or mutagenize an entire species in a very rapid manner. It's an incredibly powerful and potentially risky technology. And I've always been interested in whether anticrispers can be used to reverse a gene drive or reverse a catastrophe. And it was a group in London showed that this worked in mosquitoes and it works in yeast, and that's very cool. And there have been a handful of papers showing that in mammalian cells, and even a paper in mice from Eric Sonheimer showing that in fact anticrispr proteins can be used to endow tissue specificity for editing machineries. Been been shown to reduce off-target editing by limiting the time that the Cas9 enzyme is active in cells. And even been shown in the lab to be very useful as a toggle switch to turn CRISPR or dead CRISPR applications on or off. So yeah, absolutely. These are small proteins. They work great in human cells sometimes, and they can be super useful. And I think there's a lot of exciting applications to come. My favorite is the mouse experiment that they used anticrispers to direct Cas9 to only edit in the liver and not edit anywhere else. And it looked really nice in mice. So I think that's just fantastic.
Yay, this gets exciting. Anna, you have a question, please?
Hi. Well, I have, I was talking with you some Fridays ago, and then what we saw in Porphyromonas gingivalis, which is a pathovirus related with periodontitis, is that we haven't found any phage, right? But what we saw is that when you have some of the species of Porphyromonas in progression to periodontitis are expressing CRISPR's Cas proteins, and the spacers don't match to phages. And then what we are found now is the CRISPR in Porphyromonas is regulated virulence. So when we knock out some of the genes of the caste system, you get more virulent PG. So you see another, uses that bacteria are using CRISPR to modulate their virulence, which is very nice.
Agreed. The best example of that I'm aware of came from Francisella a few years ago. There was a paper that showed that sort of natural CRISPR-I was happening, meaning that the CRISPR machinery can be repurposed as a transcriptional repressor. And I think that was the conclusion from that story, that Francisella was repressing a virulence gene, and when you affect virulence when you delete the CRISPR system. And there have now been a couple of other regulatory stories or examples of sort of cryptic or small RNAs being used as guides, even though they're not real CRISPR RNAs. So I think that's such an exciting finding. The alternative functions for CRISPR, I think, are still being revealed. It likely is, its real job is anti-phage and anti-horizontal gene transfer. But once you've got, actually, when I started my lab, the first grant I wrote was all about alternative functions. We didn't end up going down that road too much. But if you think about the CRISPR array, it's a series of spacing sequences that turn into small RNAs that then guide the nuclease. You could easily think of this as a library of small RNAs that bacteria can build and evolve and adapt. And if you think of it that way, there's certainly a lot of potential for regulatory and alternative functions.
Yeah, definitely. And we saw in PG that there is no intracellular. That is like Francisella, it's expressing when it's inside the cells.
Cool.
Which makes more interesting. Yeah,
yes, I agree. Congratulations. That's very nice.
Thank you.
So now I'm going to turn back the attention on phages. You know that I am a big phage therapy. I'm a phage therapy proponent, full on. Yeah. And so, one of these things that I find super interesting is the acquisition or the possibility that these bacteria that are causing infection can actually have been infected at some point with phages that have allowed them to acquire anticrispers, and I'm wondering how that would impact phage therapy. Do you have,
Do you mean, it sounded like what you were saying, that the bacteria were lysogenic and had anticrispers in their genome. Is that what you were saying?
Correct, yeah. So I don't know that, so here's the thing. I know that we are trying to do a huge effort in characterizing all of the phages that we are trying to put on people.
Sure.
And that I find that absolutely amazing. The thing that we're not necessarily doing is looking at the bacteria that are infecting the people, or the bacteria that we're trying to attack, other than putting it in a plate and trying to get phages against it. And I'm wondering how having anticrispr proteins would impact the effectiveness of a phage therapy, either short or long term, specifically because we're trying to get these phages that infect the bacteria. And you can try to think that the bacteria would evolve given the pressure and how it would evolve, but still you're putting these phages that you don't know have an anti-CRISPR system in it. And maybe those bacteria can acquire also a way of silencing these phages. I don't know. I'm trying to figure out how this would impact. Because I think at the end of the day, we're looking at it as a one-hit situation where we get phages, but we're not looking at the impact of the phages and the bacteria.
Yeah, so it is very complicated. I mean, the presence of prophages in genomes of bacteria that you would like to kill is absolutely a roadblock, because probably not because of anticrispr proteins, because that seems like that would only help your therapeutic phage, but probably because of a litany of superinfection exclusion potential, and it's absolutely possible that temperate prophages can exclude obligately lytic phages that one might use as drugs. And we see that when you do host range profiling. What people often don't do is try to understand why resistant strains are resistant. They just search for a new phage, and people tend to select for sensitive strains that become resistant to figure out how they became resistant. I'm much more interested in strains that are starting out resistant, how they accomplish that feat. And prophages are a big part of that. So are a handful of immune systems. CRISPR is just one of the immune systems. It's a common immune system in bacteria, but it's not the most common. Restriction modification systems and toxin antitoxin systems by far are more dominant and probably present a bigger barrier. So despite it being my life's work, I would not say that anticrispr proteins are the be-all end-all of any phage therapeutic. I think in hosts where CRISPR is particularly active, of course, adding a protein that inhibits CRISPR makes sense. But in Pseudomonas, for example, and in Listeria, most of the strains are already encoding anticrispr proteins in their prophages, and therefore CRISPR is already inactivated. So what you really have to worry about then are other immune systems and receptors and all the other stuff we think about. So I think it's just one piece of the puzzle when it comes to phage therapy. This is why many people look for naturally broad host range phages, and what we're realizing in the lab is that these lytic phages that have broad host range, for example, like the jumbo phages that affect Pseudomonas, have very broad host range, and we think it's because they are intrinsically resistant to restriction and CRISPR-Cas cutting by the mechanisms by which they replicate, like I was saying before. Either in that case of that phage, it builds a proteinaceous border around its DNA, like a nucleus, and other phages we're working on have modified bases or have, we think, interesting repair mechanisms that make them resist any CRISPR system that cuts the DNA, not just having an inhibitor for one or an inhibitor for restriction enzymes. It has mechanisms that are more broadly acting. That's where I think, from a phage therapeutic perspective, that's where I think our work has the most impact, is defining phages that are intrinsically resistant to CRISPR, and restriction will be really useful, and the mechanisms by which they do that may be comparable to other phages through engineering. Does that answer your question?
Yes, that's just awesome. I was actually going to bring up your phages and nuclei, but there you go. You already did that.
Yeah, great. But I think
please,
that's
the
most interesting to me recently. It has not just been
love
single
that
anticrispers, but it's been that story, but also generally, we've seen a handful of lytic phages we work with in the lab, when we infect Pseudomonas with them, and we endow Pseudomonas with CRISPR immunity with a handful of different immune systems, the phage doesn't care about any of it. So some of these lytic phages are just incredibly resilient when we try to target them, and I think that's super interesting, and they all have different mechanisms to do it. So that's what we've been focusing on a lot lately.
Before I continue, you already know I talk a lot, so please, people, if you have questions, raise your hand, come up, and on that end, I'll give the floor to Bob. Hi, Bob.
So yeah, I just wanted to add on, I guess, two quick things. Pseudomonas is more my wheelhouse. Those huge phages seem to have a property that I think makes them pretty inherently resistant to CRISPRs, and the nucleus-like phenomenon that happens, where the phage DNA is physically separated from the rest of the cytoplasm for these phages, which you would imagine would make them pretty inherently resistant. And then another thing that I is sort of interesting to think about. It's, I guess, an evolutionary point, whereby being resistant to having this kind of resistance to both CRISPR and also other DNA targeting restriction mechanisms. These phages would then be able to rely on so many of their hosts being sensitive to them, which would then allow them to count on being able to infect so many more, just in terms of being able to inject their DNA into so many more hosts and rely on being able to actually infect them, allowing them to target receptors that will then be more broad. So it's sort of a two-step process where by having such a broad host range in terms of just susceptibility, they'd then be able to have a broad host range in terms of targeting of receptors.
Mm-hmm.
So I think that question is maybe solved with some of the work that you, at least the jumbo phage work that you have done, and looking at how restriction happens. Because I love that story, by the way.
Yeah. So the first point about the jumbo phages resisting CRISPR and RM with the nucleus structure, yes, I agree with that. We've been working on it for about 7 years, and we've got one paper to show for it so far. But this is a big area of interest for us, and a lot of aspects of that phage. I think there's a lot more that it does that will be very cool. The idea about intrinsic resistance to targeting by nucleases that allows them to access different receptors, I think it's an interesting idea. Those phages use the jumbo phages. We think the literature says they use the type 4 pilus as a receptor. We think that's wrong, or at least partially wrong. We see them accessing other receptors that acquire. Sometimes the type 4 pilus doesn't seem to matter. But the idea, though, I'm not sure, like even a narrow host range phage that, let's say, is sensitive to CRISPR targeting, wouldn't it still benefit from accessing broad receptor space? So I'm not sure about the idea that there would be. It seems like there's a benefit for any phage to access more broadly exposed receptors. And the question is just whether those are good receptors and whether the host can lose them or not, to take it from there.
Well, I guess there's both. Well, there's a benefit for any phage, but there's also a cost for phages that will be sensitive to a wide variety of restriction mechanisms.
I see. Yes, I see what you mean.
Then be injecting their DNA into hosts that will then restrict them.
That are not receptive. Yep.
Yes.
Yep.
So then that would be active suicide. That would then be a significant cost.
I see. So you stick with a narrow receptor range because you know what's on the inside waiting for you, and you can deal with that specific thing, and don't reach out too far because we haven't figured out how to deal with these other systems and these hosts that have other receptors. Yeah, exactly. I like that idea.
My hypothesis.
Very cool. Yeah, I think it's a great idea.
Anybody has more questions?
Yeah, I did have a quick basic question. I was wondering, when the CRISPR regions of the bacterial genomes are analyzed, do they exhibit any tropism towards certain regions in the phage genome?
Yeah, that's a cool question. There's a variety of answers on this. I can think of 3 ways to answer this question. One is the answer is no. In some cases, the spacers match all over the phage. And experimentally, you can find experiments that show that you get spacers acquired from different locations. The second answer is that yes, it is biased. If there's a so-called priming spacer, that means if there's an existing spacer that matches a phage in a specific region, but the phage has mutated itself, there's this fascinating mechanism called priming that allows the CRISPR machinery to search left and search right around that priming mismatch spacer, and therefore all the new spacers will come from the neighborhood of the initial one. And so if you were to look at a spacer data set, you might see clustering, and that's probably because there was a priming spacer that enabled more efficient spacer acquisition. And the third answer is that the Marafini lab
has shown, at least in some gram-positive phages like in Staphylococcus, that there is spacer acquisition preferably goes for the exposed injected ends of the phage genome, and they showed beautifully that that is actually during injection. And this would matter obviously for phages that always inject the same sequence first, then this would have a big impact. Maybe this is a driver for phages that have headful packaging mechanisms that inject different pieces of DNA every time. Every genome has a different topology, so maybe that's the driver there. But there is preferential acquisition from exposed DNA ends that are generated during injection or generated by restriction cutting or other sorts of DNA breaking mechanisms that happen in the cell. So I hope that answers your question. The summary is that it can be random, it can be clustered, and it can be preferentially near exposed ends.
Oh yeah, that was very thorough. Thank you.
Then Jean-Paul.
Okay, thank you. Now it's more, I think, a comment on the question. So today, the use of anti-CRISPR in phage therapy seems to be rather limited. But let's say in the future, phage therapy is common, and there would be selection for very potent CRISPR systems. And also we see in the industry, in the food industry, that they are CRISPRizing bacteria, so making them virtually insensitive to phages, because of course they want to protect their food production. And some of them, for instance, like streptococci, they also are relevant for human phage therapy. So in the future, maybe we might use anti-CRISPR systems in phage therapy.
Yeah, speaking of the future, in addition to liking your work overall, I really liked your phage therapy in the future article. That was one of my favorite phage reads of the last year. I think it is a lot of fun to think about what this will all look like in the future. The idea of CRISPR being used industrially for food products absolutely is true. I mean, this is where CRISPR was first discovered, and the first patents in CRISPR were for making yogurt. And so we're certainly, CRISPRizing bacteria. If that were to transfer into pathogenic isolates, or maybe it already has, then I agree, you could certainly see this become a problem. I think whether anti-CRISPR proteins are used or whether we simply select for phages that can already resist CRISPR based on their evolution over time, I think either way could work. The ability to, if you knew a phage therapy was failing or would fail due to a CRISPR system, the ability to add just a single small gene to the genome of that phage absolutely makes sense to me. So I think the ability to engineer these genes into phages is very simple. We do it a lot. Or to find phages that have already evolved with this pressure. I think both are logical strategies, and we'll see which ones are needed in the future, I suppose.
I'm curious about whether those strains that have CRISPR would they be able to acquire drug resistance? So would they be resistant to antibiotics?
There's nothing is an absolute. There, in some microbes where this has really been looked at, like Enterococcus, there's certainly a negative correlation. The presence of CRISPR-Cas systems is negatively correlated with the presence of islands and elements that encode antibiotic resistance. Does that mean that any Enterococcus with CRISPR is sensitive to all antibiotics? No, but it does mean that CRISPR sort of trends to exclude mobile elements, and then we found, for example, mobile elements in Enterococcus that encode anticrispr proteins to presumably get around that sort of pressure. So it is probably true that CRISPR will generally exclude foreign elements that could make the cell stronger, especially under antibiotic selection. But if that selection is strong enough, I think the cell will choose to lose CRISPR and pick up the element and survive the drug. So this is all very dynamic, as bacterial genomes are. And, no absolutes here, but that trend has been seen in a handful of bacteria. I think in Pseudomonas also there was a recent paper about it, and there seemed to be exclusion happening.
I have a question about the nature of anti-CRISPR Cas systems. might already know the answer to this, but anyway, I'm gonna go with it. So it makes sense, that these CRISPR-Cas systems already are expressed at some level in the cell, so that the anti-CRISPR Cas systems exist in a they function in a post translational manner with respect to the Cas proteins,
yeah.
right? Guide RNA loading or DNA binding. Do you expect to find any, or is it known if there are any anti-CRISPR-Cas systems that work to restrict transcription or translation of those Cas proteins?
Yeah, totally. That's a great question. We had another paper from Adair last year in Nature Micro showed that we're actually working on CRISPR regulation endogenously, and sort of serendipitously ended up finding a repressor protein that the cell uses to repress CRISPR, and we found homologs of it encoded on a handful of bacteriophages and plasmids in Pseudomonas. This is called AMRZ. It's known as a regulator of motility and biofilm production. And we did show that although these proteins do not work as traditional anticrispers, i.e. they can't inhibit CRISPR like you were asking about, when we made prophages that encoded these proteins, they indeed did repress CRISPR transcription. They turned down CRISPR. And so we proposed these were sort of anti-CRISPRs that were transcriptional repressors as one model. And then we're also working a lot on anti-CRISPR proteins that seem to impair biogenesis of the CRISPR-Cas enzymes, which I think is what you're asking about, that block transcription translation. We have an example in a more Moraxella phage, or Cas12. We think we found an anti-CRISPR protein that interferes with transcription and translation of the Cas12 effector. And it doesn't seem to inhibit it directly in vitro, but it seems to need to be in the cell and mess with transcription translation. So yeah, the answer to your question, yes, I think these strategies are out there. And what's really interesting is we think they're often paired up with a real inhibitor. So if you were a phage that could inject, you could make a real inhibitor that is made quickly and has a high affinity binder for the Cas protein to inhibit it. And then maybe you have a second protein that's there for blocking biogenesis or decreasing expression, so that when that phage is integrated as a prophage, it won't run the risk of the CRISPR system accidentally cleaving the genome. It will inhibit it and turn it off or turn it down. And we had a similar story in Listeria where we showed that the phages relied on, like this, one protein to inhibit and another protein to clear Cas9 protein from Listeria to help stabilize the prophage. So I absolutely think there's sort of anti-CRISPR synergy that utilizes different mechanisms, different points along the central dogma to inhibit CRISPR machinery.
Wow, yeah, that's a very cool two-factor system. Thank you.
Exactly, yeah. Thanks for the question.
Anybody else have more questions? Please raise your hand, come up. There are no wrong questions or bad questions at all. Anyway. Anybody else have more questions? Or I can continue asking. See, Elle has questions. There you go.
Hello. Hello, can you hear me? Yes. Yes. Hello, everyone. Thanks for the discussions, Joe and everyone. It was really interesting. I have two questions for Joe. Not so much about science, but rather about career. And I was wondering, you said that you almost directly switched from PhD to PI, basically. So how did you manage this transition? And then the second question, you said you were involved in this outreach in bioscience. So I was wondering what's your role there, and how can you manage this on the side of the lab, and what do you get from this experience as more on the startup side and rather than academia?
Yeah, that's two great questions. So starting the lab was a lot of fun because I worked in the lab all the time. I didn't really feel like I was running anything. I just, with Adair and our technician Bardo, and then Lena joined shortly after as a new student. It was kind of just a small group, and we got to work together every day, and I really loved that time. We now have 14 or 15 people in the lab, although I was just doing experiments this morning because I decided I wanted to, and so I'm doing it. But the running of the lab was more, the really made it more smooth was having colleagues around that were very supportive. I found my environment at UCSF to be very supportive, and people were helpful to lend their time and equipment and ideas. And so I think that's super important. And then I was very careful and very slow about hiring new people to join the group. I wasn't at all eager to expand rapidly. I really wanted to work closely with a few people who I enjoyed working with every day, and who we could do fun science together. So I think that the problems that new labs can have is having people join who mess up the community or the culture or the feel of the lab in the interest of growing more quickly. So I think starting out slow and as a supervisor being heavily engaged with everything that's going on, I really liked that at the start, and then had to learn. The hard part was learning how to be more of a manager as the lab grew, and that took a lot of support from people around me, and a lot of communication with people in the lab about where I know that I am deficient. And then the second question about Acrogen, it's been a long process to get that off the ground. The key part there was finding someone to run it, an experienced business and scientist who would be the CEO and run the company. I sort of am involved in the science, about a little less than a day a week, and it's physically located not too far from the lab. It's in Berkeley, it's been. I love some aspects of it. I love the nature of sort of delegation that comes with industry. I mean, like in academia, you're sort of expected to want to embrace and excel at every aspect of a project. You're expected to say, I want to do that, I want to do that, I want to do that. And because you need to learn everything to sort of do it all yourself. In industry, I enjoy the idea that you hire people who have different expertise and you form a team where not everyone has to do everything and you can move quicker together. I like that a lot. I don't like all the business jargon and drug development jargon and BS that people talk about to sound very fluffy and smart. I think there's a lot of misleading conversations, but I really enjoy the science, and I love the idea that as CRISPR has been pulled out of the phage arms race, it could really help medicine and help people. I'm amazed by the potential that small proteins that we and others have found could be useful there, and so I've enjoyed that. Without having any real expertise in the sort of drug development myself. I hope that answers the question.
Thanks for the answer.
Yeah, my pleasure.
Are you guys going to come to Evergreen, Joe? Is anybody coming? Oh, I know Jingwen is coming.
Is she? Good.
I know Jingwen is at least she submitted an abstract, so I hope she is.
Great. Yeah, I sent it to the lab. I told everyone to join virtually or in person. I wish it was. I think I was there 2 years ago, and I won't be there in person this year, but I'll definitely call in. That meeting and the Madison Phage meeting, I think it's just one day this week, and they're always at the same time. Sorry, one day this year, and they're always at the same time. So I look forward to both.
So the Madison meeting is actually only one day, but Evergreen starts right after that. So
yes,
you can attend both. There you go. Does anybody else have any more questions? I end up asking a bunch of questions usually.
I have another question if the
go ahead.
The group's fine with it. What I was wondering, I'm trying to figure out how to phrase this question. How broadly effective are CRISPR regions at blocking an infection from similar phages? That sort of makes sense. That the CRISPR region has strong homology with a number of other phages, so it can be broadly effective against those two. What is known about those types of CRISPR spacers?
The spacers that get enriched probably end up being sequenced. Probably there are spacers that come and go in
days, and they're not very effective, and they're probably spacers that stick around for a long time, and maybe those are more likely to be seen by us. Our analysis of this in Pseudomonas has been what you might expect, which is that we find that spacers that persist tend to match conserved essential regions of phages that are more likely to be homologous between related phages. So I think that's sort of what you're hinting at. But we have to keep in mind that you could have two genes and two phages that are encoding essentially the same protein. Let's say the protein is 80% identical. We'd call that a homologous protein. We'd say those phages are probably using that protein for the same function. But if a protein that's 80% identical could easily have 50 or 60% identity at the nucleotide level, which is plenty diverse to escape CRISPR. It's very easy to escape CRISPR spacers, even with single point mutations. Hell, you could have two phages with 100% identical proteins, and they could have different DNA sequences, right? So I think the prediction is true that spacers tend to match conserved stuff, because it's just kind of how the sampling bias might work out, and those might be functionally better. But I think if you just look at the sequence, it's easy for phages to escape with mutation. And then CRISPR picks up new spacers near the first one. And if those are all in sort of conserved regions, you might expect that to work a little better. But I think spacers are sort of transient. I think they come and they go and they cycle. And CRISPR picks up new spacers and the phage co-evolves. It's a pretty dynamic process. And nothing sticks around for too long. I hope that answered your question.
Oh, yeah, sure. Yeah, it definitely does. Thank you.
My pleasure.
Anybody else have more questions? You can raise your hand and come up. There you go. You can come up again now.
Yes, thanks. Yes, another question, this time more science related. I was wondering, the Anticrispr field is relatively new, less than 10 years. According to you, what are the outstanding questions which are left to be answered, and the big things which are not known yet about Anticrispers?
Yeah, that's a great question. A couple things I'm really interested in are anticrispr enzymes. So phage-encoded proteins that inhibit CRISPR but that have enzymatic function. We've discovered a couple of those that are already out there and published that have various enzymatic activities. I love the idea of finding novel enzymatic functions encoded by phages. I think we all know phages are the most diverse and exciting place to find new proteins. I would love to extend that to new anti-CRISPR enzyme functions. For example, new chemistries that could be performed by these proteins. I think how the host responds to be inhibited CRISPR is very interesting. There's an emerging theme in the world of anti-phage immune systems, which is that we all know about abortive systems where cells kill themselves when phages infect, but there's an emerging theme mostly from the Sorek lab examples where hosts, bacteria, trigger some sort of abortive response when another process gets inhibited. I think this is super cool. So there's the idea that there's an immune system that is guarding a certain host process. I would love if that such a thing exists with CRISPR, that there are backup immune systems that guard CRISPR, and that when an anti-CRISPR acts, that there is a response. Also, whether that happens for transcriptional responses to anti-CRISPRs and so on. So I love the sort of more, phage biology-centric questions around the response to anti-CRISPRs and how the cell fights back. So those are a couple of things that are in my mind. We're also working on some anti-CRISPRs that seem to have incredibly broad reach, inhibiting totally unrelated CRISPR-Cas systems. And I'm very curious how some of those work and whether they can lead us to new things we don't understand about the host, maybe. For example, if there's host proteins that are required for CRISPR to work, and the phage could inhibit those, then it could have broad-reaching implications that it leads to the inactivation of multiple totally distinct CRISPR-Cas systems. So I guess enzymes, host response, and broad spectrum inhibitors are my answers for now.
Okay, thank you.
Well, we've gone an hour, and it doesn't seem like an hour because there's still a bunch of questions. I had a question on the down low over here, asking about the sizes of the genomes of phages that have anti-CRISPR proteins. Are they all gigantic T4-like or even more gigantic genomes, or have you found they're small? Small is?
Do you still hear me? I just got a phone call. You still hear me?
Yeah.
Yeah. Typically on just your standard, temperate phage size, 40 to 60 kb sort of phages. We're actually working on a new lytic phage family, a podophage that has an inhibitor of a different immune system that I will not yet name. And there are also sort of smallish lytic phages, 60, 70 kb. I don't know, and even the ones from Sylvain, I think are on lytic phages that are not huge. So I couldn't say for sure that I know of any examples of anticrispers being encoded by any of the really big phages like the 200 kb plus phage families. Like I said before, I think those sorts of phages rely on other mechanisms to deal with CRISPR and restriction. So the answer is generally on the sort of average temperate size.
Okay. Answered question. Thank you for the person who sent it on the down low. If anybody else, this is the last question because we're almost at the top of the hour. If anybody else has any more questions, and if not, we're going to close this Phage Friday. Thanking you, Joe, for coming. Thank you so much.
My pleasure.
Yes, thank you.
Thank you.
And we're gonna do a Wednesday.
Yeah, we're gonna do a Phage Wednesday. There's another person that wants to come up and ask questions.
Oh, okay.
Come up.
One more question.
I don't know.
Hi, I just wanted to ask, are you guys going, can you hear me?
Yes.
All right. Are you guys going to be recording these sessions and podcasting them or something? I keep wanting to come and I can only catch 15 minutes of this meeting because I got a meeting prior to this. And this is such a cool topic, you guys always have really, really great talks.
I recorded this one. I will try to record each one.
All right. That was it. I just wanted to ask about that.
So we're actually going to try and see if we could do this Phage Wednesday because everything has to start with a PH.
Yeah.
Phage Wednesday on Green Room, which is a new voice app that is hosted by Spotify. So it seems that app has better way to record. So for the talks that we want to record, maybe do a Phage Wednesday and then Phage Fridays, continue to do them on Clubhouse. We don't know yet, but it's going to be our first try to our incursion on Green Room, whatever. Yeah, Green Room. Hopefully this Wednesday. And we didn't talk about the Phage printer. The Phage printer.
We can print Anticrisper.
Let's use Anticrispers to make our phages better when we phage print them in our printer for space. Yeah, it's a for those who don't know, it's a joke. We had one of our first talks was about phages in space, and we proposed that there should be a phage printer named Phage Printer with a PH, and that could print phages for different kinds of uses, but especially therapeutic uses in space. So we didn't talk about it today, but we could use Anticrispers to make our super phages in the Phage Printer better.
Yeah. And Wednesday we're going to be talking about Phage Australia.
Yep. So we hope to see there in our Phage Wednesday in Green Room. And for now, we hope to see you all next Phage Friday in Clubhouse. Do we know what we're going to talk about?
No.
Mystery again. We'll let you know through our social media.
Anticrispr.
That's what's next.
Yes.
Okay.
Okay.
All right. Bye, everybody.
It is awesome to see you all. Thank you, Joe. Thank you again for joining. Thank you, everybody, for joining. Let's see each other next Phage Friday. Ciao.